Alright, hello everyone. This is a video covering histology for the Bio 210 and 201 courses at Grand Canyon University. We have two different PowerPoints that we're going to kind of get through in this video. We're going to get through the epithelial as well as the connective tissue. So let's go ahead and kind of jump right into it. So go ahead and flip that real quick. There we go. Alright, so first up, when looking at levels of organizations, if you guys are in the 201 classrooms, you know we've talked about cells before and macromolecules and the organelles of the cells in the previous lab. And then if you're in Bio 210, this should have been an entire course of Bio 181 kind of dedicated to these macromolecules and how everything is kind of working together in a singularity to kind of contribute to this one living cell right about here. So what we're going to be doing today is we're going to actually be expanding upon that idea by talking about how these cells all work together collectively to form tissues. And this is what the study of histology is. It's the study of tissues. And tissues are ultimately what are going to create our organs. But in order to understand our organs and our organ systems, we need to get down to the basics about how our tissues maintain and function with one another. So first off, we have four main tissue types. We have epithelial, connective, muscle, as well as nervous. Now, muscle tissue and nervous tissue is not something we're really going to be covering for this week. Muscle tissue we will come back to later. Nervous tissue, we don't necessarily really come back to it, but easy way to kind of describe muscle tissue and nervous tissue. Muscle tissue is having to deal with kind of movement. So any kind of excitable and contractile tissue is muscle tissue. So this is any kind of movement that's generated in the body, whether it's conscious or not. And then nervous tissue is just dealing with the nerves that are actually branching off of your spinal cord in order to kind of initiate an action potential and kind of generate some kind of movement or action to kind of take place. But what we really need to focus on is epithelial and connective tissue. This is going to be our bread and butter for this video. So taking a look at epithelial tissue, now we have a nice little definition set out right here, but something that I actually like to describe just a tiny bit better is I like to describe epithelial tissue as the functional aspect of an organ, or known as the parenchyma. Basically, I like to think of the epithelium as, okay, this is the functional aspect of the organ. Whenever I think of an organ and I think of what that organ is capable of doing, that is usually pretty much always the epithelium. That's going to be the epithelial tissue that's actually capable of doing that. So if you're thinking about the stomach and you're thinking about how your stomach helps you digest food, well, it digests food through gastric acid, and that gastric acid is created by the epithelium. Your thyroid gland controls your metabolism. It does that through hormones, and it does that, like, it contributes to the hormones through the epithelium. So you can see that epithelium is a pretty important class when we're talking about it. And then we look at connective tissue. Another name that we can call connective tissue is called the stroma. And what stroma basically means from the Latin is it's representing support. So the stroma is our support. So the connective tissue doesn't necessarily contribute to the functioning of the organ, but rather it helps suspend it, cushion it, protect it, and pretty much everything else that the organ needs to be able to withstand a ever-changing environment. Now, with regard to how our cell junctions are actually basically how these cells are communicating with one another, we have three different methods of that, or how these cells are held together per se. So for our first one, we have gap junctions. Now, when you think of a cell, you automatically can tell that a cell doesn't have eyes, doesn't have ears, doesn't have a mouth. So that means that it makes it really hard for a cell to communicate like how we would normally. Instead, they found the next best thing. They have created these gap junctions, which are going to send chemical messengers back and forth in different kinds of ions to kind of transfuse back and forth against these little tubules. These gap junctions are these little tubules that allow for these different ions and chemical messengers to bounce back and forth, so that all of the cells are on the same page. All of the cells are know what's going on, and they know what each other cell is doing and what they're capable of. Good example of why this might be important and why we want all of our cells to be working together is because inside of the heart, right? Inside of our heart, we want a rhythm, right? We don't want our heart to just spontaneously start, doing stuff randomly. We want our heart to be able to beat and contract all at the same time. And that's up to the individual cells using gap junctions to kind of communicate that. Otherwise, we would have all kinds of arrhythmia and regurgitation of blood and all kinds of problematic functions. Taking a look at desmosomes. So desmosomes can often be represented as kind of like a fishing hook or like a J hook. That's often how it's described in lecture. Basically, these two little hooks of J's are kind of like snagged onto each other. And even though these hooks are pretty much like keeping the cells stuck together, they do still allow for some bending and some permeability. So they allow the cells to kind of expand outward if they need to, and still kind of move around. They're still very mobile like this. Another good example where you might find this in the heart is, or that's exactly what I meant to say. Another good example is inside the heart, where the heart needs to constantly contract and expand, and then this desmosomes allow the cells to do that without losing their identity and without breaking off from each other. And then finally down here we have tight junctions. So tight junctions represents the strictest adherence to each other for the cells. Tight junctions do not want to let anything through. They don't let solutes through, they don't let water through, they don't want anything to slip through the cracks. And you might be wondering, okay, well, why is this important? Well, inside of the stomach, we have gastric acid. And that gastric acid, its pH is about 2. That's very acidic, and we don't want that acidity to kind of interact with anywhere else in the environment. So this gastric acid is extremely corrosive, so we don't want it to kind of get out at all. So we have these tight junctions instead in place to kind of prevent that from coming out. These next 3 slides are pretty much just kind of talking about everything we just talked about. Alright, looking at the epithelium. So, first 2 things to talk about for the epithelium is the apical surface and the basal layer, or the basal surface. This is including basal lateral surfaces, which is like the lateral side of the cell. But whenever you're, let's say the cell is a house. So if this cell was a house, our apical surface would be like the adduct, right? They both start with A, apical surface, and adduct. And then down here we have a basolateral surface that's in contact with our basement membrane, right? So when you have a house, you have the adduct in the top and you have the basement in the bottom. Now, let's go ahead and kind of point out some of the things that we have on the apical surface. We have microvilli on this side, for one example, and we have cilia on the other. Now, let's go ahead and talk about microvilli first. So microvilli, as you can see, is a bunch of tiny little projections here. And if we actually translate the word villi, villi means fingers. So we have a bunch of tiny fingers kind of all sticking out all along the cell. And what that purpose serves is it's actually going to increase our surface area, which allows for a lot of easier absorption of whatever's kind of going on in the environment, so that these cells are more capable of absorbing anything that's kind of bypassing them. On the other hand, the cilia is more prone to moving a product in the environment. So the biggest example I can think of is inside of your respiratory tract, we have a lot of epithelial cells with cilia on them. And this cilia actually allows us to move mucus up the respiratory tract. So the cilia is more interactive with the environment and kind of moving products along that are outside of the cells. A way that I like to kind of think of it as an analogy is crowd surfing. So if you go to a rock concert and there's like a lot of crowd surfing, you have fans in the crowd that are all moving their hands back and forth, like picking up the rock star and moving them along. Well, that's kind of like crowd surfing. These cilia are all like those fans' hands, kind of moving that product about. Now, for this next thing real quick, I'm going to go ahead and go over to paint, just because PowerPoint is a little finicky with regards to how it accepts my drawings. So let's go ahead and draw something out right like this. So what I'm doing right now is I'm going to draw out a few different cells, right? So we got a few different cells here. Got 3 different cells on top. They all got their own nuclei. We got some little microvilli kind of coming out of the bottom here. And then we got another set of cells right about here. Now, I promise in class I'm normally a much better artist with a whiteboard and marker, But drawing with the mouse makes things a little bit more complicated. Like, look at that. I don't even know if that's a nucleus. That just looks really messed up. There we go. Now, as we can see, the way that the cells are facing each other, they're kind of creating this open space in the middle. Now, this open space in the middle is known as the lumen. Oh, jeez. Even writing words is harder. So, the lumen exists as a hollow space inside of an organ or inside of a vessel. That is our biggest or best definition of what a lumen is. So, good example is if you're eating something, it travels down your throat into your stomach. The actual area that that food now exists in, that chyme, it exists inside the lumen of the stomach. So any organ that you have that's hollow, that can actually be filled up with something, this is known as the lumen. So the lumen of the heart would be filled up with blood. Lumen of the gallbladder is filled with bile. And another good example is actually if you take a look at a blood vessel, something like this, right? And you got your little blood cells kind of traveling through your little blood vessel. The blood cells that are actually traveling through your blood vessel, they are traveling within the lumen of the vessel. So the lumen is just the hollowness inside. Now you might be wondering, okay, well, why did I just talk about this lumen for like the past like few minutes? Why is it so important? Well, because previously we described that, okay, the top of the cell is the apical surface, right? And then the bottom side is the basal surface, and that's connected to the basement membrane. And that was pretty nice and easy. But cells inside of our body are kind of like astronauts in space. There's not necessarily a correct up or down. Instead, we all base our positions on the relativeness of something that we all have in common. And something that the cells have in common here is actually the lumen. So, the lumen is gonna be what determines our apical surface. So when we're looking at our cells over here, this is now our apical surface. And on the opposing side would be the basal surface. So you can kind of see why it's important to kind of understand and differentiate where the lumen is, because that's going to determine the surface of the cells, and then the opposite side is going to be our basal surface. Alright, let's see. Yep, once again, basal surface, apical surface. This is all stuff that we love and know already. Now, when we're dealing with a little bit more with regards to our epithelium, we have a few different ways we can categorize it. So, at the top, we have two different rows. We have simple and we have stratified. So, simple is referring to the fact that it is only a single layer. So, all of these cells only have one single layer. So, counting from the lumen down to the basement membrane, there's only one nucleus, right? Only one nucleus. But taking a look at stratified represents anything that's more than one. So anything that is more than one will be considered stratified, multiple layers on top of one another. So that kind of gives us a definition for these rows, but let's talk about these columns. Now these columns are actually based on our shapes, the shape of the cells, and what they do to exist. So if we're looking at squamous cells here, right? Our squamous cells, whenever I think of squamous cells, I think of squished, right? That's the first thing that comes to mind, because this thing looks like it just got like hammered into the floor here. All of these cells, like, they look like they're really trying to defy gravity, or there's some kind of like blobfish monster pulled up from the depths of the ocean or something. They're all like kind of pushed in and very squished. Taking a look at cuboidal, the first thing you should have thought of when you saw cuboidal is cube, right? It's very square-like, very cube-like. Obviously, these images are 2D, but these are 3D cells. So these are full-on cubes. So these are cuboidal, stratified, cuboidal, simple. And then columnar. Columnar is dealing with rectangles or columns. So all of these columns are basically stacked on top of one another. So here we can see simple squamous epithelium. Yes, this is good to point out that you do have to write out the entire name for your quiz and exam. So please make sure that you're actually writing out the full description, simple squamous epithelium. Because if you negate even one of these words, we have no idea what you're talking about. Now, taking a look, we have a lot of different locations and functions for our various epithelial shapes and layers. So taking a look here, we want to think about the characteristics of what makes it simple squamous epithelium. Now, if we look at simple squamous epithelium compared to every other type of epithelium, this is easily the thinnest layer. There's no going about it. This is the thinnest layer that we could possibly come up with. And the reason why we might want it super thin is for permeability. And permeability is referring to the fact that we can have oxygen as well as carbon dioxide easily pass through the cell to travel into a new environment. So you want to think about the lungs in this case, or the alveoli of the lungs. This is the biggest example where you might think that these guys are coming from. So inside of the alveoli of the lungs, this is where we have an exchange of oxygen and carbon dioxide with the environment. So an alveoli of the lungs, that's going to be super important to kind of get this gas across without any need for any kind of energy to bypass the cells. It can easily just diffuse across. This also is going to occur inside of vessels as well, inside of various tissues. This is actually what's going to allow us to deliver oxygen and nutrients to various tissues throughout the body too. There's just more examples of that. I'll make a separate video just going over just pure examples for you guys. So stratified squamous epithelium, right? Many layers, squished epithelium, right? Now, stratified squamous epithelium is primary function protection, right? That's what you want to think. It's protection against abrasion, which just means bruising, pathogens, referring to any kind of bacteria or virus, and then chemical attacks as well. Hopefully you're not being chemically attacked, but no worries. Stratified squamous is there to protect you. So you want to think about the most exposed parts of your body with regards to what might need protection. Surface of the skin is your immediate go-to. Your skin is in contact with everything in the environment, so obviously it's going to need a lot of extra protection. Now down here we can actually see that our stratified squamous has two subcategories. It can be keratinized or it can be non-keratinized. So the only thing keratinized and non-keratinized is differentiating is it's telling us whether or not these layers of cells have keratin. And keratin is essentially a structural protein that's going to provide even more protection than what our stratified squamous is already doing. Now, unfortunately, we can only kind of keep this in one general location. We're going to keep keratinized stratified squamous on the skin because that's obviously where we're going to need the most protection. One little note too is that whenever we do have a lot of keratin filling up these cells, the cells are actually going to be killed off. So if we take a look at the next slide, we can see down here we have our nice little cells, and we can tell that they're alive because they all have the little nuclei, right? But once we get to this thick layer of dead skin, they no longer have nuclei, which means that these are dead cells, which means that we only have one possible answer if we were seeing this on a quiz or exam. This would be our keratinized stratified squamous epithelium. Something else I forgot to mention too when we're generally talking about this is your general game plan for whenever you're identifying epithelium is kind of coming in three parts. The first thing you're going to do is you're going to find the lumen. The second thing you're going to do is you're going to find the basement membrane, right? So lumen, basement membrane. And then the third thing you want to do is you want to count the nuclei from the lumen down to the basement membrane. And that's going to tell you simple or stratified right away. The other thing you're going to want to do is kind of look at the shape. And the shapes are usually pretty easy to kind of differentiate. Taking a look at this guy compared to the previous slide, this is our non-keratinized stratified squamous epithelium. So you can see there's no dead layer of skin cells all the way at the top. Looking at simple cuboidal epithelium, now whenever you're thinking of simple cuboidal epithelium, you're always thinking secretion and absorption. And you might be thinking, okay, well, secretion and absorption of what? The biggest example that you're going to find is inside of the kidneys. So your kidneys are in charge of what is kept inside of your body and what is urinated out. And that's done through a whole different level of mechanisms that we'll get to much later on in our anatomy knowledge. But for now, simple cuboidals are actually what are committing to secreting and absorbing various aspects of the kidney. So whenever the kidney decides, oh, we need to keep some of that sodium around, or we need to keep some of that potassium, that would be the absorption with that regard. Or we need to get rid of some of these hydrogen ions, that would be the secretion. So our simple cuboidal is just involved in a lot of secretion and absorption. Taking a look here, we have stratified cuboidal epithelium. Now, stratified cuboidal epithelium, this is a little bit more of a rare type. This is primarily found in your sweat glands. Here we can see multiple lumens. So here's one lumen, here's another lumen, a third lumen. Now don't let these multiple lumens kind of confuse you. We're going to follow the same game plan. We're going to find the lumen, we're going to find the basement membrane where the cells typically end, and we're going to kind of circle in around it, and then we can count multiple nuclei stacked on top of each other in this case. Transitional epithelium. So, when we think of transitional epithelium, we want to think of transitioning. And you might be wondering, okay, well, what kind of transitioning? Now, when we're looking at the epithelium for transitional, it's more of a process of elimination. Now, we can't exactly categorize this as anything but transitional epithelium because it doesn't have that squished appearance, because you have some cells down here that kind of look squamous, but then all these other cells are kind of like ballooned up. We can't be cuboidal because while some are cuboidal, others are very circular. And then columnar is just kind of out of the question entirely. Transitional epithelium has its uses. It permits a lot of expansion and recoil from stretching. So very, very useful in general. Especially inside of the urinary bladder. That's like your primary example. Your urinary bladder is going to fill up with urine from various parts of your day, and the transitional epithelium is going to be what is going to allow for that expansion. Here we're looking at simple columnar epithelium. So simple one layer, columnar is referring to the column shape. Now the general region that this simple columnar is going to be found, it's going to be found along your GI tract. So your gastrointestinal tract is often where you find the simple columnar epithelium, like your stomach, like your intestines, your gallbladder. Now the size and shape is going to allow for, once again, secretion and absorption and partially some protection. Most cells have the protection, so that doesn't often help us narrow it down, so I don't often include it in my descriptions. But these simple columnar, they're huge, so they increase a lot of surface area, which is really good in the digestive tract because we want to absorb a lot of nutrients. And then in the gastric stomach, in the stomach, we can actually secrete a lot of that gastric acid through our simple columnar epithelium. Here looking at stratified columnar epithelium. Whenever you see stratified columnar or stratified squamous, you're always thinking immediately just protection. This is just multiple extra layers that are going to allow us to protect our areas better. And once again, this is primarily in areas that are exposed to the outside world. This is a fun one. So this is also another category that's kind of like transitional epithelium, doesn't really fit in with anybody else. So here we see pseudostratified ciliated columnar epithelium. Now the reason why we call it ciliated is because the pseudostratified is always ciliated. So that's a nice and easy way to kind of differentiate it from other epithelium when you guys are looking at different examples. So when we look at this example, pseudo. Pseudo means fake, and that's what it translates to. So pseudostratified means fake stratified. You might be wondering, okay, well, why are we calling it fake stratified? It looks pretty stratified to me. Now, every cell has an apical and basal surface. We know this, right? And when every cell has its apical surface reach the lumen and its basal surface reach the basement membrane, that's considered simple. And in this case, this is exactly what's going on. Our pseudostratified ciliated columnar epithelium is in contact with the lumen and the basement membrane for every single cell that we're kind of seeing across. So even though it looks like multiple cells are stacked on top of one another, this is actually all considered only one layer. That's why we're going to call it pseudo-stratified. And biggest area we're going to find it in is inside of our respiratory tract. So inside of the respiratory tract, as well as some portions of the reproductive tract, this is where our mucus with the cilia from that example I provided earlier is primarily going to occur. So all that mucus is going to get moved up from the cilia as the cilia interacts with the environment. So, taking a look at glands. Excuse me. Now we have two different types of glands here. We have an endocrine gland and we have an exocrine gland. Now for the basic definition, an endocrine gland is something that is ductless. An exocrine gland is something that contains a duct. Now there's actually an easier way that I kind of like to break up these glands when I'm dealing with them. So let's go ahead and take a look at the word endo. Endo means within. So endocrine is referring to within the body in this case. So don't want to confuse anybody. Crine doesn't actually refer to body. Crine refers to like hormones and products. But in this case, endocrine is referring to staying within the body. So that's why we don't really need a duct with endocrine. That's why it's considered a ductless gland, because all the products that it is creating are meant to stay within the body. And they're going to do that by circulating around in the blood. So we can see right here, secretes into blood. And it's going to allow our products to kind of circulate around the blood and stay within the body. If we look at the word exo, exo refers to outside. So, an easy example I like to think of is of an ant. The anatomy of an ant has an exoskeleton. So an ant with an exoskeleton has a skeleton on the outside of the body. And that's kind of what exocrine is going to be referring to here. Exo is referring to creating a product that is going to travel outside of the body. And that's actually why we need this duct. We need this duct because we're traveling outside the body into a potentially dangerous environment. So we need like a special route to get there. Good examples of exocrine glands include your sweat glands, right? That's all the sweat that's produced on the outside of your body. Mammary glands can also be included for milk production from the mammaries. And then we also have saliva, right? Whenever saliva is created within the body, it's actually going into our mouth, which would be considered outside the body because it's technically more exposed to the outside world now. It's not within the bloodstream anymore. These are just examples of the major endocrine glands. This is going to be a huge topic in 202 and 211, but for now, we'll kind of just leave it at this. Here we can see exocrine glands, the different examples that we already talked about. With regard to simple and compound glands, we're just going to skip this. We don't really cover this in my class. Finally, we have our exocrine glands and how they're classified by secretion. So we have three different types of secretion. We have merocrine, apocrine, and holocrine secretion. Now, merocrine secretion is the most basic. So we have a cell here. The cell is going to create a product inside, right? That's eventually going to travel into a duct. And these small little products are just going to use our Golgi apparatus to just kind of disperse into the environment, into the duct. So it's going to be small little bits of secretion. Here we can see apocrine secretion. He is obviously way more dedicated to the cause. So apocrine secretion is a lot more invested, I guess you can say. So he's actually willing to pinch off a large portion of himself and secrete it as a product into the environment. So he's actually going to branch off a huge component of himself and send that off as a product. And then holocrine is easily the most dedicated soldier. He kills his entire self. Now obviously these cells can be very regenerative. That's how we can continue to use our exocrine glands, but these mature cells, they die and they themselves become the secretory product. So yeah, that's basic categorization of exocrine glands. Alright, so that pretty much concludes this first video for epithelium. At first I wanted to do kind of them side by side, but I feel like breaking it up into two parts is going to be a little bit easier. So stay tuned for connective tissue.